The objective of this scientific video article is to review biochar literature, specifically biochar feedstocks, production methods, characterization, modification, and potential applications.
Biochar is a stable carbon-rich product prepared from biomaterials through various heating techniques available in our
Biochar provides several environmental benefits, and the most important include its potential to mitigate greenhouse gas emissions and improve soil health. Recently, biochar has also been used as a precursor to prepare catalyst electrodes of fuel cells and as a supercapacitator, as well as in soil and water remediation. Biochar production.
Biochar is usually produced through methods such as pyrolysis, gasification, torrefaction, flash and hydrothermal carbonization. Of this, pyrolysis is the most common and widely used method to produce biochar.
Biochar can be produced through pyrolysis using a simple muffle furnace or microwave system in a laboratory to batch kilns, or a continuous feed pyrolyser for large-scale production. In most cases, continuous mode reactors are preferred, and typical ones include rotary kilns, fluidized bed and fixed pyrolysers, and auger or screw-type pyrolysers. These type of reactors involve continuous feedstock input, as well as output of pyrolysis products such as bio-oil, biochar, and syngas. Thus, continuous mode often results in high operational efficiencies, as well as a higher biochar yield compared to batch modes. Here we explain small-scale production of biochar. A muffle furnace with modifications to collect syngas and supply nitrogen were used. Sample plant materials were placed inside the furnace. Nitrogen gas was fed at a fixed flow rate to remove air inside the furnace. After closing the furnace, pyrolysis, temperature, and time were set. During pyrolysis, the organic components are thermally decomposed into a vapor phase, while the residual solid phase remains as biochar. After the process, the furnace is allowed to cool down. Then biochar is taken out. The vapor phase collected is then cooled to produce the bio-oil, in which polar and high molecular weight compounds are condensed, while the low molecular weight volatile compounds, such as carbon monoxide, hydrogen, and methane, remain in the gas phase. Pyrolysis can primarily be divided into the following methods: slow and fast pyrolysis. Fast pyrolysis is generally described as the thermal decomposition of biomass in the absence of oxygen at medium temperatures, around 400 to 600 degrees centigrade, using a very high rate of heating, usually greater than 300 degrees centigrade per minute, and a short residence time, less than 1 hour. It is generally estimated that through fast pyrolysis, a low yield of 10 to 30% biochar and 15 to 20% syngas, a very high yield of 50 to 70% bio-oil can be expected. On the other hand, slow pyrolysis is defined as carbonization of the biomass over a wider temperature range, 300 to 800 degrees centigrade, with a low heating rate, 5 to 7 degrees centigrade per minute, and a long residence time, usually more than 1 hour. Slow pyrolysis often results in the high yield of biochar, typically 35% biochar, followed by 30% bio-oil and 35% syngas. Biochar feedstocks.
Biochar are usually procured from biomaterial, which are usually regarded as waste, are available in plenty, or obtained at low cost.
In particular, feedstocks of biochar production are often derived from agricultural and forest residues. Most of these resources are regarded as byproducts or even wastes of harvesting and subsequent processing of crops. Some important biochar feedstocks include coconut shell, rice husk, bamboo, grasses, pine wood, sugarcane bagasse, seaweed waste, municipal sludge, pig manure,
The production, properties and yield of biochar strongly influenced by time and specific properties of feedstock as well as pyrolysis conditions such as time and temperature.
Mechanism of biochar production.
Biochar feedstocks are generally composed of cellulose, hemicellulose and lignin. During pyrolysis, each component is decomposed at different rates through different mechanisms and pathways.
Biochar feedstocks are generally composed of lignin, hemicelluloses, and cellulose, and each component is decomposed at different rates through different mechanisms and pathways during pyrolysis. The rate and degree of decomposition of these three constituents depends primarily on the process parameters such as temperature, reactor type, heating rates, and feedstock particle size. For example, during pyrolysis, the hemicelluloses first decomposes in the temperature range of 200 to 260 degrees centigrade, whereas cellulose breaks down in the range of 240 to 350 degrees centigrade. And finally, the decomposition of lignin begins at temperatures from 280 to 500 degrees centigrade. In the pyrolysis process, cellulose is initially depolymerized into oligosaccharides, followed by the cleavage of the glucosidic bond to produce D-glucopyranose. This in turn undergoes an intramolecular rearrangement to form levoglucosan, which in turn converts to levoglucosanone through a dehydration process. Levoglucosanone can then proceed through several pathways, including dehydration, decarboxylation, aromatization, and intramolecular condensation to form solid biochar. Modification of biochar.
Although biochar have several beneficial properties to suit various applications, they can be further modified to enhance its efficiency.
The modification of biochar can be accomplished through physical modification and chemical modification. Important physical modification methods include steam activation. With the aid of the steam activation process, synthesized biochar could be converted to activated biochar with high surface area and enhanced carbonaceous structure. Gas purging. Carbon dioxide modification alters biochar surface by creating microporous structures, as carbon dioxide could react with carbon of the biochar sample to form carbon monoxide. Important chemical modification methods include acid or base modification. Using chemical agents such as hydrochloric, sulfuric, and nitric acids, or sodium and potassium hydroxide, modulation of surface acidities and porous structure of biochar can be achieved. Functional group modification. Chemical oxidation using potassium permanganate, nitric acid, phosphoric acid, and hydrogen peroxide can introduce acidic binding sites such as carbonyl, phenolic, lactonic, and carboxylic groups on the biochar surface at relatively low temperatures. Biochar nanocomposite. Metal oxide biochar nanocomposites are expected to perform well in adsorption compared to raw biochar owing to the presence of two solid phases, including a biochar matrix and nanocrystals of metal oxides. Both of these phases are expected to contribute to the overall adsorption capacity of composites.
This scientific video review summarizes recent advances in the field of biochar research. We have highlighted various potential feedstocks, their modification methods, biochar synthesis, and their characteristics as well as potential applications of biochar in agricultural activities. In general, the utilization of biochar as sustainable and economic high-value product for various applications appears to have a huge scope in near future.